First, thanks for having me. It's great to have a community of people trying to build in this space who are all aligned around this mission of letting us live longer, healthier, and longer. And BioAge, it's been a decade now, right? So it's been a long journey. It's been really interesting to watch the space change through time. And the original founding vision was pretty simple. It was that, biology is really hard. Everything's incredibly complicated. How can we get more certainty about which drug targets are going to matter and translate into actual human studies? And you look around and you see there's data sets like Decode Genetics or UK Biobank that pharma are using to de-risk drug discovery. Because if there's a human signal for a gene, it's more likely to work in a trial. And we wanted to build basically the aging version of those kinds of biobanks. So what we do with the company is we work with a lot of biobanks that are like UK Biobank. If you're familiar with that, it's like a population resource with blood and genetic and health information from a huge chunk of the population, basically. But with one key difference that makes it more relevant to aging. We were looking for biobanks that started to collect sample from middle-aged people when they were healthy 50 years ago. And the reason we wanted that was because we wanted to look at those blood samples that were collected and see which molecular features, like the proteome or the metabolome, could predict disease, could predict different lifespan trajectories over time. So really building out these data sets to get conviction around which targets are relevant to human aging. And that's really stayed the core of the company through time. So we have multiple programs now that are based on targets that have a signal in those human data sets. One of them going into the clinic later this year, and collaborations with a couple of companies, pharma companies, on earlier stage discovery from our platform. So that's something we still continue to invest in.
Yeah, okay, cool. So let's dig a little bit deeper into this. So you're looking at data from humans in these biobank samples. So what do you find? Like, what are the interesting targets? How are they involved in aging?
Yeah, you see a lot of classic stuff that's well known, like a lot of inflammatory factors that go up as you get older. And people who are like middle-aged, for example, who have lower levels of those tend to live longer, tend to be functional longer. We also try to look for those cohorts where they have not only simple information about how long you live, but also like how did your muscle function, how did your brain function change through time. So there's a lot of classic ones, I would say, that are well known. Some of them are more suitable as like biomarkers than drug targets. Like GDF15, for example, is a beautiful biomarker that we see a huge signal for across all our cohorts. And then some of them are more novel, right? Because for those of you, I think a lot of you are working in this space, but part of the motivation here too is that aging is still in many ways a new science. Like a lot of the targets we frankly don't know which ones are going to work yet. So I think it's important. For us, it was really important to do new discovery as well and find new targets that hadn't been discovered yet. And if you look at the field, like one of the best papers from last year was a totally new target, IL-11, right? Making mice live longer. So we also wanted to do totally de novo discovery of novel targets by leveraging these new platforms, right? Like today, we work with a company called SomaLogic that can go into a blood sample and measure like 11,000 different proteins, right? And some of them are known to be involved in disease processes, but many of them are not. And you can do that discovery for the first time from these kinds of human data sets.
Right. Yeah, so I want to tap into, this, this expansion of data and what's out there potentially, and different tools that we could use to enrich data and so forth, but and collect it. But I guess maybe we can go a little bit into some of the insights in these targets. So you guys have a pipeline right now. Maybe you can discuss a little bit why you're so excited about like your lead program right now.
Yeah, so our most advanced program today is an NLRP3 inhibitor. This is a part of the inflammasome. So your body becomes and your blood becomes a lot more inflamed as you get older. There's a lot of inflammatory factors. Even if you're very healthy, even if you eat very well, it's still going to happen. It's part of aging. And what's true of many aging targets is that these changes predispose you not just to one, but to several different diseases, right? So if you have higher levels of NLRP3 signaling pathway, if you are more inflamed at an older age, you're more likely to get Alzheimer's disease, but also cardiovascular disease, right? So these targets that are relevant to multiple different diseases. And so we built a very potent NLRP3 inhibitor that gets into the brain, that gets into the eye, relevant potentially for many different aging-related diseases. It's a target, too, that someone has shown, if you knock it down, can make mice live longer. Very small study, but very nice results, very sort of encouraging results. And we should have some initial phase 1 data from that compound later this year.
Yeah. Wow. Yeah, I didn't know about that lifespan data. Yeah.
It's not like a powered study, but it's still suggestive, like aging cell.
Yeah. Okay, interesting. Very interesting. Okay, so yeah, circling back to data. So you mentioned you guys are doing proteomics with SomaLogic. Now, the general vibe in the space, we're in the Bay Area, is like AI and data, right? So like, let's talk about the data stuff. So like, you have SomaLogic. What else are you guys measuring? I mean, you have these biobank samples, so you can probably go back and use technologies that are coming online today. So what's on your radar in terms of like the kinds of stuff that you want to capture data-wise? Yeah,
that's a great question. I mean, for these samples, we've historically done proteomics, but also metabolomics. And even of those two, like from a drug discovery perspective, proteomics is so much easier because if you find out what the protein is, well, chances are someone has done some work on that protein before. You're not starting from total scratch, right? So while there are other technologies like methylation, which are really good for biomarkers, it can be a tough starting point for drug discovery. So I do think there's a lot of information on those other technologies, but maybe best suited initially at an academic stage. We're still doing metabolites too, but even that is already harder, right? That's already like a harder level of explanation. So we're big believers in the proteome. And this 11,000, I mean, BioAge has been around 10 years now. And the first time we did one of our biobanks with SomaLogic, it wasn't that long ago. It was like maybe 6 years ago, 7 years ago. That was 4,000 proteins. So even these sort of more routine technologies are changing rapidly in just the past few years. And it's really integrating that. I mean, we use these data sets to find out what's specifically important for aging populations, but then you can go and integrate it, of course, with all the other genetic information from these big public resources like UK Biobank. So you want to plug it into that whole system as well. So that's one part of the kinds of data analysis, AI type analyses that we do, but it's very small data compared to, what LLMs are usually crunching, for example, right? That said, we also like to leverage everything else that exists in the world, right? we want to convince ourselves it'll translate to humans because it has that nice human signal in our own data set, but we also want to leverage all of known biology. So one effort that we are doing initially internally to sort of leverage this kind of information is to build sort of more custom LLM-style approaches to extract information from scientific literature and present it in a way that can help our scientists really prioritize targets. That is being led internally by Kia. So if you want to learn more about that, go talk to Kia. We are planning to open source some of that at some point too. So we think these tools would be more broadly useful outside the company. Thank you.
Awesome. Yeah. There's a lot of, I don't know, when you look in the news, especially news about AI, like just recently, I think Demis Hassabis, who's the founder and I guess CEO of DeepMind, right? He made a comment that he believes that all diseases will be solved within 10 years. Potentially, I'm not an expert in AI, so, but there is this sense that on the AI side, there's like this vibe of people saying, we're going to solve biology really quickly. But I'm just curious, like for someone who's on the ground, right, dealing with biology, wrestling with biology all the time, trying to develop new medicines. For people, definitely in the context of aging, like, is the AI mostly hype, or do you see like new AI tools like really being high leverage for the work you guys do at BioAge, or do you see something bright on the horizon that's coming?
Yeah, for sure. And, I would love it, of course. It'd be amazing if we could cure all diseases in 10 years. That would be the best thing ever. Biology is hard, right? Like, I think there's certain areas of the drug discovery funnel where AI is having a huge impact today, right? And like one is like in helping us to discover new and better molecules, suggest new molecules. Because these are ones where we have large amounts of training data, or AlphaFold, right? Because you have the protein structures that you can train on. You have large amounts of training data. You can leverage that to do a lot of steps where you can go and test and get your feedback really quickly. Like that's the kind of setup you need, like lots of existing data and the ability to iterate quickly to really leverage these new tools. So I think that at the beginning of the funnel where you're building novel molecules and where you're doing target ID in the first place, there's a lot of like very specific areas where it'll definitely accelerate things. Biology's hard though, right? The challenge in drug discovery has always been you might have a molecule and it like works amazing in a mouse and then it doesn't in a human, right? And we can't simulate human biology. partly because we can't get the right data. You can't sort of like destructively sample a person every minute and like model that. That's going to remain a challenge for some time. So that's not to say that this isn't going to have a huge impact. It will. Like, I think a lot more molecules will get, more rapidly developed to that initial stage. I think you can learn a lot in initial clinical trials, and that'll have a positive impact. But I think that'll still remain a hard piece for data acquisition reasons.